Hey guys, today we are going to review the cell membrane and then we're going to talk about different ways that things can pass through the membrane. While you are taking notes on this, make sure you have the vocab list out so you can get a definition for every single vocab word. Some of them will be on the slides, but some of them I'm just going to say, so you need to be paying really close attention. And make sure you're taking additional notes. Shouldn't just be a list of vocab words when you finish. All right, we're going to start by reviewing the cell membrane. So we talked about the cell membrane way back in standard 2 or 1, one of the two, I forget. So let's review it so that we are all refreshed in the cell membrane. So remember, the cell membrane, we called it like the bouncer of the cell, right? So it can control what can come through and what cannot come through the cell. And remember, all cells have cell membranes. They all need this bouncer. And the cell membrane is made up of a lipid bilayer. So a lipid bilayer are two layers of lipids. And remember, these lipids have this hydrophobic head and the hydrophilic tail, and they go two rows together, right, to make a waterproof barrier. And so this is why it is able to let some things through and other things can't come through. And you can see that in the picture. So again, you have this lipid bilayer, two layers of lipids. The outside are hydrophilic, water-loving. The insides are hydrophobic, water-fearing, and this gives you that waterproof barrier. And this is why the cell membranes are selectively permeable, or what we call semi-permeable. So it means the same thing, right? It means some things can get through, other things can't. So if you look at that picture there you can see the red guys can move in and out, but the white triangles cannot get through. So that's our cell membrane, right? We also called it a fluid mosaic model. So if you look at these images, hopefully that will give you an idea of why we call it a fluid mosaic model. So again, the cell membrane is made up of all of these lipids, right? All these heads and tails throughout, but it also has these proteins embedded within it. So that's why we call it the lipid bilayer, or that's why we call it the fluid mosaic model. So fluid, because it's moving around, right? Look at that little guy. It's shaking and moving around. Those proteins can float here and there. And mosaic, because again, it's made up of lots of little pieces that together make the big piece. So here are just some pictures of mosaics. And again, you can see they're small tiles or small images that together make up the full picture. So again, cell membranes, right? They're the bouncer of the cell. All cells have them. They're made of a lipid bilayer, which are waterproof, makes them semi-permeable or selectively permeable, and we call it the fluid mosaic model. Good. All right, now that we've reviewed, let's talk more about cell transport, so how things move around through the cell membrane. And we're going to start with diffusion. So diffusion happens when you have two sides of your membrane and one side has a lot of stuff in it and the other side doesn't have a lot of stuff in it. And diffusion is when that stuff that's in the water or in whatever liquid we're looking at will move from areas of high concentration to low. So it's going to move from where there's a lot of them to where there's a little of them. And this will continue until eventually they become balanced, which we call homeostasis, or equilibrium will be the term we use here. Okay, so diffusion, stuff moves from high to low until it's balanced out. A specific type of diffusion is called facilitated diffusion. So some things can just diffuse right through the membrane, no problem, like in this picture, just moves right through. Other stuff needs help to get through. So facilitated diffusion is when stuff is going to get helped to move through that cell. So you can see there, this one, it goes through a protein. So it can't move through on its own. It's too big, but it can move through a protein. That's facilitated diffusion. And then there's also active diffusion. So active diffusion requires energy, while the rest of diffusion just happens on its own. Active diffusion requires energy. So a lot of times it'll go in reverse. So it can take things from low to high if you want a really high concentration. But it's just one that requires energy. So when we talk about diffusion, I'm going to go back a few slides here. Some words you need to know are solute and solvent. Okay, so the solute is what gets dissolved in whatever you're looking at. So in this picture, the solute are these red spots, right? It's whatever's dissolved in. So it could be salt or sugar or proteins, whatever it is, it's dissolved in. Solvent is what the rest of it is, right? It's what's doing the dissolving. So usually the solvent is water. It's called the universal solvent because it can dissolve just about anything. But it's what, has the solutes in it. So in this picture, it would just be this liquid. This is probably water, would be the solvent. So again, solute, what's in it, solvent, what it's in. So if we get technical here with diffusion, diffusion is where your solute is going to move from high concentration to low concentration. And again, in this picture, the solute is the box. The solvent is whatever is outside and inside the cell, water in this case. And again, same thing here, your solute are these Na pluses and these Ka pluses, and the solvent is water. Okay, and the important thing to remember about diffusion is that it's the solute that's moving. So with diffusion, the solvent, the water stays the same because the solute can move in and move out. All right, this is different than osmosis, a different type of transport. So osmosis is when water moves through the semipermeable membrane. So this is when the solvent moves. So osmosis will happen if your solute can't get through that membrane. So if the solute can't move, then the water will move and we'll have osmosis. So if we see in this picture, we have a solute here, it's this green guy, right? Our solvent is water. And you can see these solvent pieces are really big. They can't get through that membrane, right? Look, they're too big to fit through. So what's going to happen is the water is going to move. So if you look at the water levels before and the water levels after, you can see that water moved from where there were low concentration of solutes to where there's high concentration of solutes. So water moved through the membrane towards the direction where there's lots of solutes. So water is going to move where there's not a lot to where there is a lot. And we're going to use scientific terms for not a lot, lots, which are hypotonic versus hypertonic. So hypotonic and hypertonic are comparison terms. So you have to use them together. It'd be like saying, Bob has more pizza. More than what, right? So it's a comparison term like more and less. So hypotonic means less solute, right? So if you look at the outside of this cell, there's less solute than there is over here. So hypotonic is less solute, right? There's only one versus a bunch. So you would compare hypotonic to hypertonic, right? So hypo, less solutes, hypertonic, more solutes. And then isotonic, this means they're equal. So if I look at both sides of the membrane here, they're equal out. That is an isotonic solution. So with an isotonic solution, the water is just going to move back and forth freely, but it'll stay the same overall. So if we looked at like a tubing like this one, the water would be the same level and it would stay the same level the whole time. If your tubing had one side being hypotonic and one being hypertonic, that's when you would actually see the water move. So if we look at each of these cells, these are the animal cells at the top. Again, if it's in an isotonic solution, so if outside is equal to inside, water will move in and out, no problem. And that's normal in an animal cell. In a plant cell, that's actually not great. That's when the plant's going to be kind of droopy and sad. All right, if your cell goes into a hypotonic solution, all right, so a hypotonic solution, remember that means less on the outside, right? So now you just have a few solutes on the outside, lots on the inside. Water is going to move into the cell. So again, water moves from where it's hypotonic to where it's hypertonic. So it's moving into the cell. For animal cells, this is bad news bear. The cell will burst. For plant cells, this is normal. So water is moving in and that plant cell gets nice and full, which is what you want. If the cell is in a hypertonic solution, the opposite will happen. So hypertonic means there's lots of solutes outside, and it means the inside of the cell is now hypotonic. So the inside's hypotonic, the outside is hypertonic, so water is going to move out of the cell. And you can see this will cause the animal cell to shrivel, and the plant cell is going to shrivel as well. You can see it's shriveled within that cell wall. Cell wall stays, but the cell inside will shrivel up. So again, water is going to move from where it's hypotonic to where it's hypertonic. So water is always going to go hypo to hyper. Okay. The last type of movement is when something is either taken into the cell or expelled out. So endocytosis is when the cell sends, or excuse me, when the cell takes stuff in, endo, bringing it in, endocytosis. And there's two types of endocytosis. One is phagocytosis. And with phagocytosis, the cell's going to take in like a solid particle. So what it does is it takes its little plasma membrane, it reaches up, it scoops, and it sucks it in. And that's a vesicle or a vacuole. Penocytosis is where it's going to take in fluid. So again, that cell membrane will come up, it'll surround the fluid, and it'll take it into a vesicle or a vacuole. Exocytosis is the opposite. Exocytosis is where the cell's going to release something. So this is when a vesicle will come up to the membrane, it'll join up to the membrane here, and then it'll expel out whatever it is. So this could be waste product, it could be a signal for another cell, but it's just another way stuff can move through the cell membrane. All right, so make sure you have all your vocab done, and check in with Miss Taylor when you're finished. Good luck studying, guys.